Hey, let's talk about fighting viruses, shall we? It's me, Moser, and after this video, you should be able to distinguish between vaccines and antiviral drugs as means of fighting viruses. You should be able to analyze what new ways of creating vaccines mean for COVID-19 vaccine progress and describe the ways in which common antiviral drugs work. This all assumes that you know the difference between viruses and bacteria, as well as just a little tiny bit about basic viral structure and function. Okay, let's go. You know what viruses are, and that they use the cells of their living hosts to reproduce themselves, instead of doing it themselves. Ta-da! There's an animal cell just chock full of viruses, bursting open to release them. You also know that antibiotics, which act on the life processes of bacterial cells, are useless against viruses. So how do we fight viruses? Well, there are two major tactics that humans use against viruses. Vaccines as a prevention strategy to stop viruses before they can infect a human, and antiviral drugs, which can be used once someone is infected with a virus to slow or stop the progress of the disease. Now, you already know a little bit about how vaccines work to help your immune system defeat viruses before they can get a foothold. Let's look at the process of developing vaccines for the novel coronavirus of 2019, SARS-CoV-2, and how that's proceeded. First, you got to understand that the current virus troubling humanity is only one of many so-called coronaviruses. Corona means crown, and these are all viruses that have spikes topped with little proteins sticking out from them. So they look like they're wearing a little viral crown. Viruses in this family, well, they include the 229E alpha CoV, the NL63 alpha CoV. Wait a minute. You've never heard of these. Right. We tend to only hear of a virus when it's big trouble. And many coronaviruses, like the ones I just listed, well, they're pathogens that just cause minor diseases. Think like the common cold. But a few coronaviruses have made the big time in past decades. And, that includes the SARS-CoV virus, the first one, which caused a major disease outbreak in China in 2002, and the MERS-CoV virus, which caused a major disease outbreak in the Middle East in 2012. These waves of emerging disease meant that after the big outbreak of Ebola virus, that was a nasty one, in a couple of West African countries, in 2016, the World Health Organization put together a list of the viruses that were most likely to cause serious disease outbreaks in the next decade. Sort of a most wanted watch list kind of thing. And the SARS-CoV, which had been seen in China in 2002 and 2003, though it was last seen in 2004, well, it was on that list. So researchers have done lots of work with coronaviruses, a lot of them in the past, and as a result, they know a lot about them. For instance, remember those spike proteins? Well, they know that the spike protein is the piece that coronaviruses use to unlock a human cell. Well, if a human body can make some sort of antibody, a chemical which hooks onto the spike protein, it's kind of like coding a key to a lock in bubble gum. Now the key no longer works because that space is taken up. Researchers had already been working on a new kind of vaccine for the SARS virus, the first one. The new twist was that instead of the usual method, where a vaccine delivers either a weakened or inactivated virus or a dead virus, well, these researchers attempted to create a vaccine built to deliver a small piece of the virus's RNA, so that the cells of the person receiving the vaccine can actually build one of the proteins from the virus, and hence give their immune system something to recognize, so that they can start blocking that virus fast by creating those little proteins that hook onto the spikes. Cool. When the novel or new coronavirus, SARS-CoV-2, what we know as COVID, popped up in 2019, researchers realized that the new vaccine type they had been working on might make it possible to use the same delivery mechanism, but swap the RNA from the old virus with the RNA from the new virus. They knew that if this worked, it would significantly shorten the amount of time it took to develop the vaccine. It's kind of like baking cookies from scratch versus using a box mix. They tried an RNA-based virus for the novel coronavirus, the SARS-CoV-2 2019, and found that it was tolerated by humans. Further studies to determine what dose is needed to develop immunity are going on right now in the fall of 2020, and studies to prove how well the vaccine generates immunity are scheduled or ongoing in several countries. Other researchers have begun building similar vaccines that are based on DNA with the exact same idea. Give human cells the genetic code to build proteins. Several DNA-based vaccines have already been tested, including ones for HIV, Ebola, and the Zika virus. Why use DNA instead of RNA? Well, DNA vaccines have some advantages over RNA vaccines. For one thing, DNA is stable for a lot longer at room temperature. Think dried egg noodles. While RNA needs freezer storage. Think frozen seafood. Using this sort of modular approach to developing vaccines, kind of like the watches where you can switch out the bands on the same watch, well, it might actually be the future of vaccine development. If the crop of vaccines that are currently in development and those that have been recently developed work as well as hoped, well, it could be. But what about curing a viral infection once it's happened? That's where the antiviral drugs come in. While vaccines prepare the body's immune system to fight the virus on its own, antiviral drugs in some way disable viruses that are already in the body. Remember that when a virus invades a host, it breaks into the cells of the host and tricks the cells into making copies of the virus until the cell is full of copies of the virus, at which point the cell either just bursts or the viruses that have been created actually unlock the cell from the inside. Tamiflu, the antiviral drug sometimes used against the influenza virus, works by binding or attaching to a protein that viruses use to get the host cell open once they've been created. Then the virus can't spill out of the cell and go on to infect more cells. Remdesivir, the antiviral drug recently approved to treat COVID-19, works by attaching to the viral RNA, boop, so that when copies are made, they're damaged and they're not functional. Then those damaged strands of RNA can't be used to create new virus. So in short, most antiviral drugs work by binding or attaching to something that the virus either needs to get into or out of cells, or something that it needs to force the cell to do its replicating. Okay, do you feel able to distinguish between vaccines and antiviral drugs? Can you analyze new ways of creating vaccines and what they mean for COVID-19 vaccine progress? And can you at least briefly explain the ways in which antiviral drugs work? Great! Then go on and, hey, cover your face.